Assalamualaikum and very good morning. Today you will learn about the histology of the cerebellum. This is the learning outcome for this lecture. When you section a piece of cerebellum, it shows two regions, white matter and grey matter. Grey matter is present at the surface of cerebellum and is known as cerebellar cortex. Grey matter also present deep in the white matter as deep cerebellar nuclei. As you know, grey matter is composed of neuron cell bodies, dendrite, unmyelinated axon, and glial cells. The white matter is composed of myelinated axon and glial cells. Cerebellar cortex is divided into three layers. The outermost layer is called the molecular layer. The middle layer is called the Purkinje cell layer. This is a very thin layer. And the innermost layer is called the granular layer. So this is the molecular layer, the outermost layer. And this is the granular cell layer. And in between these two layers is the Purkinje cell layer. Molecular layer contains two types of interneuron. The first interneuron is called the stellate cells, and second interneuron is called the vascular cells. Stellate cells present mostly in the outer part of the molecular layer, whereas the vascular cells present in the inner part of the molecular layer. Besides this interneuron, you also have other structures such as dendrite of Purkinje cells, axon of granular cells, and dendrite of Golgi cells. Purkinje cell layer is a very thin layer located between molecular and granular cell layer. It is occupied only by cell body of Purkinje cells, not by the dendrite and axon of Purkinje cell, because the dendrite of Purkinje cell is located in the molecular layer, whereas the axon of Purkinje cell extends down into the granular layer. Granular cell layer is the deepest and thickest layer of cerebellar cortex. This layer contains granular cells, and it contains a lot of them in this layer. It also contains Golgi cells, unipolar branch cells, and the cerebellar glomerulus, which is a synaptic complexes. This diagram shows the different structures located in the three layers of cerebellar cortex. So this is the molecular cell layer, this is the Purkinje cell layer, this is the granular cell layer. So in the molecular layer, you can find the two types of the neuron, stellate cells and vascular cells. In the Purkinje cell layer, you can find only the cell body of the Purkinje cells. In the granular cell layer, you can find Golgi cells, granular cells, and glomerulus. Now let's talk about the neuron of the cerebellar cortex in more detail. Purkinje cells is the largest neuron in the entire central nervous system. It is a multipolar neuron with a large cell bodies and extensive dendritic trees. The cell body is flush-shaped, and it's located in the single layer between molecular and granular layer. The axon passes through the granular layer to reach the white matter of the cerebellum and terminate mainly in the deep cerebellar nuclei. Dendrites of Purkinje cell are extensively branched and extend to the molecular layer and has the shape of fan. It uses GABA as neurotransmitter, so therefore it is inhibitory neurons. Purkinje cells are the only efferent neuron or projection neuron of the cerebral cortex, because the other neuron in the cerebellum do not go beyond the cerebellar cortex. This diagram to show the outline of Purkinje cells. It has cell bodies, which is located in the Purkinje cell layer. Its dendrite is extensive and extend into the molecular layer. And it has a single axon which pass down in the white matter and synapse with the deep cerebral nuclei, and also in the vestibular nuclei. Granular cells. Granular cell is the most numerous neuron in the cerebral cortex, but it has the smallest cell bodies in the entire central nervous system. And in fact, the total number of granular cells is more than the neuron population in the entire cerebral cortex. Okay. This is the cell body, and it has quite a number of dendrites, in fact 4 or 5 in number, which form contact with mossy fibers. And granular cells use glutamate as neurotransmitter, which is excitatory neurotransmitter. Axon of granular cell pass into the molecular layer where it bifurcate into T-shaped configuration to form parallel fibers. Parallel fibers are parallel to the long axis of the folium, and they also parallel to each other. Parallel fibers run through the dendrite of series of Purkinje cells, just like the telephone wires running through the branches of series of trees. And these fibers also make contact with other neurons, such as stellate neurons, basket cells, and Golgi cells. A single parallel fibers can make contact with thousands of Purkinje cells, and only any one Purkinje cells receive synaptic contact with thousands of parallel fibers. So this is the granular cells. Granular cells. It axon extend to the molecular layer and it bifurcates into T-shaped configuration. So this is T-shaped configuration, and the parallel fibers, they are parallel to each other and they runs through dendrite of thousands of Purkinje cells. And these parallel fibers also make contact with other neurons, such as stellate neuron, basket neuron. Other neuron located in the granular layer is called Golgi cells, which is normally located adjacent to the Purkinje cell. Its dendrite extend up into the molecular layer, where it make contact with the parallel fibers. The axon branches and form synaptic contact with the dendrite of granular cells to form cerebellar glomerulus. And this neuron use GABA as neurotransmitter, so therefore this is inhibitory interneuron. This is to show the Golgi cell. The cell body of Golgi cells is much larger compared to the cell body of granular cells. It dendrite extend into the molecular layer and make contact with the parallel fibers. The axon extend down and make contact with the dendrite of granular cell to form synaptic glomerulus. Unipolar brush shell is the newly discovered cells. It is found primarily in the granular cell layer of the vermis and floccular nodular lobe, and that there are very few of the cell in the cerebellar hemispheres. This cell has slightly oval cell body. It has a single dendrite and single axon. Its dendrite is quite short and ends up at brush-like configuration called the dendriolus. So this is a brush-like configuration of dendrite called the dendriolus. This is the dendrite. The axon located in the granular cell layer, and ends as series of 2 to 4 synaptic structure called a unipolar brush cell rosette. These cells use glutamate as neurotransmitter, so therefore it is excitatory neuron. Basket cells located in the deeper part of the molecular layer next to the Purkinje cell layer. It axons synapse with the Purkinje cells, and it is called a basket cell because the axon itself forms a basket-like arrangement around the cell body of Purkinje cells. It dendrites synapse with the parallel fibers of the granule cells, and this neuron use GABA as neurotransmitter, so therefore this is a inhibitory interneuron. Stellate cells is located mostly in the superficial part of molecular layer. These cells use GABA as neurotransmitter, so therefore this is a inhibitory neurotransmitter. Its dendrite synapse with the parallel fibers. The axon synapse mainly with the Purkinje cell dendrite. So these are to show the basket cell located in the deeper part of molecular layer. As you can see here, the axon of this basket cell, it form basket-like arrangement. Okay? It form basket-like arrangement around the cell body of Purkinje cells. So that's why it's called the basket cells. The stellate cells located in the mostly in the superficial layer of the molecular cell. It has a shape of a star, so that's why it's called stellate cell. Okay. Although the basket and stellate cell are similar in general shape, the extent of dendrite and axon field is much larger in basket than stellate cells. Consequently, basket cell may influence larger number of Purkinje cells, whereas stellate cell influence a much smaller population. And both of these neurons receive excitatory input from the parallel fibers. This is the summary of the various types of neuron in the cerebral cortex. You can refer to this table and study them in more detail later. Next, we'll talk about the terminal efferent fibers in the cerebral cortex. Cerebral cortex receive input from many different sources, such as from spinal cord, vestibular system, from the inferior olivar nucleus, frontal nucleus, and many more. And in the cerebral cortex, these efferent fibers terminate as either mossy fibers, clumping fibers, or monoaminergic fibers. Climbing fibers. Climbing fibers originate from inferior olivary nucleus, and this is the only source of the climbing fibers from inferior olivary nucleus. In molecular layer, these fibers climb directly up into the Purkinje dendrite. So that's why it's called the climbing fibers, because it climb dendrite of Purkinje cells. And each Purkinje cell is innervated by a single climbing fibers, unlike the mossy fibers. And climbing fibers use aspartate as neurotransmitter, which is excitatory neurotransmitter. Mossy fibers originate from deep cerebral nucleus and other part of the brain, such as spinal cord, medulla, pons, and many more. In the granular layer, mossy fibers branch profusely, and their terminal, which is called the mossy fibers rosette, enlarge and make contact with other neurons, which gather to form the cerebellar glomerulus. And each single mossy fibers can affect thousands of granule cells. And these fibers use glutamate as neurotransmitter, which is excitatory neurotransmitter. So these are mossy fibers. You can see it form rosette, numerous rosette. So these are to compare between the climbing fibers and mossy fibers. Climbing fibers originate only from the inferior olivary nucleus. This is the only source of climbing fibers. It is called the climbing fibers because it climbs the dendrite of Purkinje cells. And one climbing fibers may contact with one Purkinje cells. Whereas Mossy fibers originate from other sources such as spinal cord, pons, midbrain, and so on. It has many sources of Mossy fibers. And these fibers, it form a rosette and make contact with granular cells. It may contact with Golgi cells to form glomerulus. And from this, the impulse carried by the Mossy fibers will be carried by the granular cells and into the parallel fibers. And Mossy fibers make contact with thousands of Purkinje cells. Cerebellar glomerulus is a symmetric complex found within the granular cells. It is formed by symmetric contact of the granular cell dendrite, Golgi cell axon, Golgi cell dendrite, and terminal segment of Mossy fibers, which is Mossy fiber rosette. Mossy fiber rosette is centrally located and form synapses with several granule cell dendrite. Golgi cell axon contact with granular cell dendrite in the glomerulus. The entire complex is encapsulated with glial cell processes. So the entire cerebral glomerulus has a form of capsule formed by the glial cell process. So these are to show the cerebral glomerulus is formed by the Mossy fiber rosette, Golgi cell dendrite, and the granular cell dendrite, and is covered by a so-called capsule formed by the glial cells. Okay. So these are cerebellar glomerulus. Monoaminergic fibers originate from cell of the locus ceruleus, raphe nucleus, hypothalamus, ventral membrane of tegmentum, and some other location. In the cerebral cortex, these fibers branch diffusely and terminate in the molecular and granular layer, where they may influence other neurons. Thank you very much for your attention.